So off my screen here. So I'm Alec Filipowski, and along with Alan Watson, we are the MPIs for the Brain Image Library project. And joining us today are team members Luke, Mariah, Ivan, and Ayana. And today we're going to tell you about the data submission process at BIL. I'm briefly going to start with a brief introduction to BIL. We're going to go on to using the submission portal. You transfer data to BIL, and there's a number of different methods that you can use to transfer data to us. And we're going to also show you how to use on demand to look at the files that you've transferred and enable you to reorganize your files if you need to. Then we're going to talk a little bit about submitting data to BIL in terms of file organization. About midway through today's session, we'll have a break. And then after the break, we'll talk about the metadata schema and how you fill out the different metadata spreadsheet depending on how your data has been generated. We'll then go over uploading your metadata to the portal. And then once everything is complete, submitting your data for validation and publication. Talk a little bit about what is involved in the validation process. And at the end of the process, you will get a DOI, and we will talk about DOIs. We do issue a couple different DOIs. One is the DOI for the data set, and the other DOI is for a collection of data. And the data set DOIs are issued by default. If you need a collection DOI to group a bunch of data sets for a paper that you're publishing, we will talk about the process that you would use to request that collection DOI. And then we'll end up with questions and individual discussion, individual help. I should mention that during this workshop, if you need help at any point in time, please make a note in the chat, and one of the BIL team members will help you with the issue that you're having. Tomorrow, we're going to be focused on using data at BIL. I'm not going to go over this agenda in much detail, but we'll talk about finding data at BIL, some of the tools that we make available, how to use Jupyter notebooks with a few different demonstrations that we have. Then we'll have a break. We'll talk extensively about the BIL Analysis Ecosystem and different ways that you can visualize data in BIL. And we'll follow up at the end of the workshop with a question and answer session and help people on an individual basis. So let's begin. So unfortunately, for those of you who've been around for a while, some of you may have known our third PI, Marcel Boucher. Marcel has unfortunately passed away last year with a long battle with glioblastoma. We truly miss him as a collaborator, and also he was a good personal friend to many of us on the BIL team. So the focus of my talk this afternoon is to provide a brief overview of BIL and the services that we offer. I'll then talk about a few of the features that we are working on that we expect to be made available over the next 6 months to a year. And then I'll end with some advice on how to get started contributing data to BIL. The Brain Image Library is a public resource serving the neuroscience community, and we provide a persistent centralized repository for brain microscopy data. The library relies on contributions from brain researchers like you, and much of our current data has come from investigators that are part of the BICCN and BICAN efforts. NIH funding is not required to contribute data, but the data must be of interest to the BRAIN Initiative. And since many of the data sets that we house at BIL are quite large, we provide an extensive analysis ecosystem where users can explore data in place without downloading it at no cost. We also provide complementary network path analysis to sites experiencing networking issues, either uploading or downloading data. The data scope for BIL includes whole and partial brain data sets from mice, primates, including humans, and model organisms, targeted microscope-enabled experiments, including connectivity between cells and spatial transcriptomics, and historical collections of value to the community. To give you a preview of some of the high-quality data in BIL, this image shows a cell population data set contributed by Dr. Pavel Austin, formerly at Cold Spring Harbor Laboratory, now at Certigo Therapeutics. This two-channel whole brain image data set is about a few hundred slices and is one of the smaller data sets that we house. Some of the largest high-resolution mouse data sets currently in BIL are generated using the FMOS technology. These FMOS data sets range from about 8 to 13 terabytes each and contain about 10 to 15,000 slices per data set. The resolution of each of these individual slices is about 35,000 pixels by 55,000 pixels with a z-spacing of about 1 micron. In the near future, we expect to have data sets that are much larger as the BICAN network is now focusing more of its data generation efforts on primate data and humans. In terms of existing coverage, the overwhelming amount of data sets that are contributed to BIL are from mice, with relatively few data sets at the current time from macaque and marmosets. In terms of data modalities, most of our primary data sets are slice-wise image stacks from whole brain imaging experiments. In addition, we have thousands of tracings from primary image data sets that have been deposited in the archive. In terms of technology, we have data from most microscope types, as well as microscopy data produced by multimodal experiments. Overall, in terms of data, the Brain Image Library has been doubling the data sets that we receive about every year and currently have over 15 million public files. Going too far here. Next, I want to briefly mention some of the additional services that we provide to data contributors. These services, as mentioned earlier, include networking path analysis for people who are having troubles with data transfer. We can also receive data via alternate media, such as tape or hard disk drive, where the networking path is insufficient. Mentioned earlier, we also issue DOIs for our data sets, and we can issue collection DOIs on request. Next, I want to briefly mention our analysis ecosystem and some of the resources that are provided through that analysis ecosystem. These resources include both small and large memory nodes, nodes with GPUs, and our Bridges 2 and Cerebrus CS2 high-performance computing systems. BIL also provides a remote desktop environment and the capability to host unique web interfaces to the data. And we also support numerous software packages as well. All of these resources are made available to both data contributors and data users at no charge. Next, I want to briefly tell you about some of the coming attractions at BIL that we expect to make available over the next year. One of which we'll talk a little bit about tomorrow is our on-demand transformer that will be capable of serving data from BIL and converted on the fly into selected formats, for example, Neuroglancer and Napari. We also are undertaking greater coordination and linkage with large consortia, including BICAN. And we're also going to be making some changes to metadata in terms of making our metadata more usable and greater accessibility, particularly with other resources. We intend to do a metadata crosswalk that compares the metadata schema that BIL uses with schemas in use elsewhere, and have a way that, if it's possible, that we can translate fields in our metadata schema to fields in somebody else's schema. We also intend to provide greater links to related resources, such as the Allen Brain atlases. Furthermore, we do intend to manage our project space and resources better through the Access program. So how should you get started contributing data to BIL? Well, in the pre-proposal stage, we encourage you to contact us through our hotline at bil-support@psc.edu and discuss your project with us a few weeks prior to your proposal submission. What we can provide for you as a part of that submission is a letter of support stating that BIL will accept your data. And we can also provide a template for those of you who are generating data with NIH funding, a template NIH data management and sharing plan outlining specific standards in use in BIL. Many of you are aware, but some may not be aware, that NIH has a new data sharing policy that requires submission of data management and sharing plans with each new proposal. So that's what we can provide pre-proposal. After your proposal is funded, or if you don't have NIH funding, and this is something that a project that you're using internal funds for, again, we encourage you to contact us at bilsupport@psc.edu to discuss your data, but this time in a little bit more detail. It would be helpful at this point in time to know the amount of data that you intend to be submitting to BIL and the time frame that you will be submitting that data, as well as what your anticipated submission time frame would be. We also, at this point in time, would like to discuss with you any specific visualization requirements that you would have for your data. I mentioned earlier that our on-demand transformer will be able to serve data in a number of different formats. And we want to make sure that if you desire to have your data visualized over the web, that we receive it in a format that we can use our on-demand transformer to visualize that data with. We will also advise you on what our current metadata template is and how to fill it out for your data, how you can structure your data when you upload your data, and some additional instructions on how you can gain access to all the BIL systems, as well as any upcoming training that we are offering. And then finally, I just wanted to make some notes about data submission, which will be the focus of the remainder of this workshop. Some good advice that I'm going to offer people who are submitting data to BIL is to please start the data submission process well in advance of any deadlines that you may have for making the data public. Part of this planning considered the time required to move the data. And again, we can provide assistance locating where transfer bottlenecks are occurring. But again, as data sets grow larger and larger, the amount of time required to move the data will increase. Once we receive the data and you're ready to submit data to us, the data goes through a validation process. And in general, the validation time will be proportional to the number of files in the submission. Generally, if it takes you a week to transfer your data here to BIL, you should expect that it would take us at least a week to validate that data. Some general rules about validation. There's no hard and fast rules here, but a smaller number of large files will generally validate quicker. And a smaller total number of data sets in a submission envelope will validate quicker. In other words, it's better to have 5 different submissions, each having 5 different data sets than one submission with 25 data sets in it. And principally the reason why is because if the larger submission runs into a problem, it's going to stop the submission process. And the things that have validated prior to that data set will not be able to be made public. Whereas if there's smaller submission packets, data that has passed validation can be made public much quicker. We also have curators, and our curation team may need your assistance to resolve issues in the validation process, as well as in your metadata. So that's all I was going to go over today. I would now like to turn the workshop over to Luke.
Just a moment, let me get my slides up here. Okay, hi everyone. My name's Luke. I'm a web developer at the PSC, and I also work closely with the curation team on data submission and the files that come into BIL, as well as handling user tickets and issues that users may have. Today, I'm going to talk about the submission process itself, on how to create a submission at BIL and get the files over here and everything you need to do to get that data public. So the workflow process of creating a submission at BIL begins with creating the submission, structuring your data, transferring that data to BIL, ultimately making that data public. But in between that, you'll need to provide metadata for the submissions. We'll get into more of metadata structure and more information about that later on. And I'll also touch on the PI dashboard, data manager dashboard that we developed for data managers and PIs to have a larger overlook of the submissions at BIL as a whole for your project. And before I go any further, I just want to ensure that everybody can log into the submission portal itself. So if folks could head over to, in their browser, to submit.brainimageLibrary.org and enter your credentials and see if we can get everybody logged in. I just want to make sure we can get there before we begin any sort of hands-on sessions. And I'll drop that link to there in the chat. And just by a show of reactions of thumbs up, if we've gotten to that point, would be great. Okay, great. We're seeing a few here. If you have any issues logging in, please feel free to send those messages in the Zoom chat channel for any help needed. I'd also like us to log into ondemand.bil.psc.edu. We'll touch on a bit more of what this is later on in the presentation. This is the same credentials you'll log in. You've logged in with in the Submit Brain Image Library website. Oops. It's like we're getting. One moment, I think I need to reopen it. There we go. So if folks can get there too, that would be great. Great, we're seeing some thumbs up there. So I'll head back over to the presentation. And we'll head into the first slide here. Most of y'all have done this already. If you haven't, I have some instructions on how to create your account at BIL. But in case folks are sending this around to other colleagues who haven't created a BIL account yet, this is some information on how to do so. So to create an account at BIL, you'll first need to create an account through the Access portal. Access is the new service we use that replaced Xseed for folks that used Xseed in the past to create their BIL accounts. Under the Brain Image Library website, there's a walkthrough on this information on how to create an account through Xseed, or sorry, rather Access, and give that information over to us to activate a BIL account. So when you go through the Access portal, there's information about your institution, your name and information like that. It'll create a username and password for you. At that point, once you've created an Access account, send over a ticket to BIL support letting us know that you created an Access account, and we'll finish setting that up for you. And then when you do your, when you've been given the green light that the account's been made, you can set your initial password at apr.psc.edu. And that'll look something like this once you create your password. And at that point, you are ready to log into BIL and create a submission. I should mention too, when you create a new account at BIL, you'll be given a default project to submit to. I'll get into this a little more later too. When you send over your information about the Access account and you're a data submitter, if you could also send along information about the grant, as in the grant name and number that you'll be submitting for, that'll allow you to be able to submit data to that project within BIL. But if you log in initially, you'll have a private Scratch space to mess around with, get a feel for how to submit things to BIL, or just have a spot where you want to deposit something to do some things with the hardware available on BIL. So as I mentioned before, the submission portal located at submit.brainimagelibrary.org. Through this portal, you can create a data submission as well as get a spot for you to deposit that data to, which is known as a landing zone space. Within the landing zone space, the only person who has access is you and us. So you can do kind of your file structure work there, or you can do that locally, whichever is more comfortable for you to do. So to do that, on the submission website, on the top bar, there's an option for new and new submission. Clicking that will get you to a form to fill out to get that landing zone directory to point your data to. So we'll go ahead and do that. So we're already at brainimageLibrary.org. I think everybody was able to get logged in there, hopefully. If not, please let us know and folks will help you out. If you're unable to at the moment, just follow along the best you can. So if we go to the submission website and click on new, submit new submission, here's where we get that form that we showed in the presentation. Just for the sake of the workshop, we can name it something like April BIL workshop. In practice, making these fields as descriptive as possible is always better. Just say not applicable for that now. And down at the bottom here where it says project, if you click this, a few options should pop up for you. Some of you that have submitted in the past should have access to projects that you've been submitting for. Otherwise, you'll have some scratch space called project 1 or new project, things like that. Just for now, we can just choose one of those. And if we hit save there, that'll bump us over to prompt us to upload a metadata spreadsheet. But we're not going to do that just yet. We'll do that later in the presentation after Mariah goes over some metadata practices. What we're more interested right now is going to view and submissions and finding that submission we just created. For me, it's right here, April BIL workshop. And if you take note, there's a field called data staging area. So this is the directory on BIL that you'll point your data upload to, which we'll do an example of later in the presentation. I want to take a moment here just to make sure everybody's on the same page. If we need to slow down or stop or anything to get everybody up to speed, feel free to unmute and say something if you do. It's a small group.
I already have a submission in progress. Should I still create a workshop submission, or can I play along with my existing submission?
It's up to you. We're just gonna do like kind of a dummy upload to a dummy submission later on.
Okay. Yeah, there's like data moving now, so
That's okay. So if you already know that, Katie, you can follow along as much as you want.
Thanks.
So with that, with the spot for us to drop our data into, we'll go back into the presentation here and show what you would do next. So now that we created that submission, like I said,
can you hold on a second? One thing that we did not mention thus far, but if you don't remember your password, there's a special website that you can go to reset it. The website is apr.psc.edu. So if you open a web browser and go directly there, that will let you reset your password.
right. Happy to wait a moment to continue if folks need to reset their passwords too.
I have a question. So I already have established a project, and do you submit to a project? I'm kind of getting confused again about the terminology.
So you established a project as in when you go to that project field in the form that item that you're talking about is available to click on?
Yeah, like it has a name, funded by, is BICCN personnel submissions. So then do you submit to the project?
Yeah, if you were ready to point a submission to a batch of data to that project you're submitting for, you would choose that in the form.
Okay, where do you choose? Where do you
think of the project as a tag that goes along with your data.
And then
So a number of the researchers involved are part of the BICCN and follow-on BICAN efforts, and the groups at the Allen Institute have established projects and sub-projects for each type of data, and so that they can categorize things on their end a little bit better. So what we do is we have people have their projects on BIL to be the same as what they would get through those consortia. Now, if you're not part of one of those consortia, it really doesn't matter what that label is. But if you are, that label then becomes important to them.
Okay, so I see when you create a new submission, you will give the name of the project will be the project that's in the that I've established. That'll be I'll fill out that field with the name of that project.
Yes.
Okay.
Yeah, and if that field, when you click on that piece in the form, if that project doesn't pop up for you, let us know and we can make sure that that does, because that's how the indexing actually happens and gets that tag.
Yeah, it looks like it does.
Okay, great.
Thanks.
Yeah, no problem. So I'll go ahead and move forward then to discuss the next step on after you've created that spot to send your data to. And that's the portion where we'll do a data transfer over to that directory. There's a lot of options available for data transfer depending on the operating system you're using and your either aversion or willingness to use a command line interface. There are many options available. For Windows, a lot of folks will use SFTP or SCP either via the command line or through the programs WinSCP, FileZilla. There's a lot of programs that do it. Those are just the most common ones that we see people use. Within this presentation, these are also clickable links for you to get to any of these if you want to try any of those out. Another really common one we see folks use is Globus. It has a very user-friendly interface to send files through. I think I have a hand up. Is that Katie?
Yeah, I'm just wondering, is Globus not supported for Linux? Or shouldn't?
I don't believe it's supported for Linux as far as I can tell.
I mean, it is, but it really depends on whether or not you have, whether or not somebody's installed it. The installation procedure is completely different. So, but by default, it's usually not installed on Linux systems.
Okay.
But you can certainly get the software and install it.
We'd probably be using the Python SDK anyway, I think so. But thank you.
Yeah, I think if you aren't using something like Ubuntu with it won't have the interface and you'd have to use the SDK. Okay,
thanks.
No problem. With Mac OS, you can also use SFTP, SCP. Mac OS does not support WinSCP, of course, as it is described in the name, but FileZilla is another good alternative to that, as well as Fetch. That's also available in the command line. And since Mac is a Unix-based system, it does allow rsync, which is a strictly command line file transfer protocol. I'll show an example of each of these as well, two of which I'll do a kind of one in practice. We'll do an SCP example later on together. Lastly, with Linux, since it's Unix, there's, of course, SFTP, SCP. There are some available interfaces depending on the distribution you're using of Linux, and rsync is available for that as well. So here's an example of actually someone using rsync to transfer a file up to BIL. As you can see, they're structuring it as the rsync command, a few flags set. They're pointing it to mouse1, which is a directory for a data set of mice that they have images for. And they're pointing it to their username at upload.brainimageLibrary.org, and then that landing zone directory we created before. That's an example of someone using rsync within Unix. I'm going to show an example of how to use Globus for folks who may be interested in that. To use Globus, you'll have to log in with your access information. There's a few links here on how to get started with Globus, as well as downloading the Globus Connect Personal application that gives you the interface to be able to do the file transfer. How it would work if you're going to use Globus is you would head on to cilogon.org, choose the institution via select a provider, and provide your access information there. Once you've done that, you'll have the information that you'll need to send over to BIL support to get the endpoint to submit to. And I'll show where that is on the next slide, but it's under the user attributes dropdown. I'll show that next here, the information you need to send in that ticket to get the endpoint. Or I can actually show it through the Globus itself. If you give me one moment. So if you were to log on to that CI login link and go to the user attributes, you'll have a certificate information here, certified subject. This is the line that you would need to send along for us to set you up with that endpoint, as well as your username. And once you have that access, within that Globus UI, you'll be able to point a link to the brain image library upload with CI login authentication. Again, I'll show this all an example here in a moment. But if you search brain image library, two options should show up for you. There's a download, which is which doesn't require any authentication because it's public data. But if you want to do that upload, you would choose that option here, brain image library upload. How one would do this file transfer within the UI of Globus would be as follows. Up on the top left, I'm looking at my local machine and I'm choosing a file called myfile.txt. And up at the top right is where I chose the brain image library upload with CI login. And below that is that directory that we created through the submission portal. It's a simple drag and drop over. It's nice because it gives you email on the status of your transfer and kind of gives you, unique identifiers to show you exactly what's going on with your data transfer. And I can show that here. In my own Globus file manager. So on the left here, I have just my local machine. And on the right, if I go up here and click that and type in brain image library, this is the option we're looking for, is this upload with CI login. You can see here that we have a few directories to go down. What's an easier thing to do here would be to when you took note of that data staging area, just copy and paste this. Actually, it's easier if you go back here and do like this. And just drop it in there. Doing that will take us to this empty directory we created. And you simply just drag and drop a file into there. And you'll see if you do that under activity, you'll see the different transfers you've done, a little more information of what happened, how long it took, the number of files transferred. I did this earlier, just transferred a file. It only took a few seconds. So if you'd like to do that, that mode of file transfer, just follow along those instructions and we can get you set up with Globus. But like I said, there's many other options we can choose. Here's an example that we're going to run later is using SCP to upload to BIL. Very similar to the rsync command, just the name of the command, the file or directory they're looking at, and where to upload it to, being that username, upload.brainimageLibrary.org, and then the directory that you created. Here's another example of a user interface file transfer application. This is WinSCP. WinSCP looks pretty similar to Globus. But again, it's specific to Windows. FileZilla looks very similar to this too. So if any of those are interesting to you, there are links in the presentation to check those out. But they all operate pretty much the same way, except with these, you wouldn't need to do a Globus endpoint. You can kind of just log in this way and it will authenticate you. So we'll do an exercise here on actually uploading a file to that directory we created. So we'll just do a quick creation of a dummy file, and we'll do this within the terminal, or you can just create a file within Notepad or something easy to use. If you want to do it through terminal on the Mac, if you go up to the top right at Spotlight Search, type in terminal and hit enter, you'll get your terminal open for you. On Windows, just the bottom left search, typing CMD and enter will open that terminal for you as well. And on Linux, Control-Alt-T will bring that open for you. So we can do this together, and I'll show the example as well. I'm just going to go up here and type in terminal. And let me make sure I'm sharing my whole screen here. So I'm just going to type CD to get to my home directory. And I'm going to run cat my new file. This will open a new file for us. I'm just going to, oh, sorry, am I moving too fast? Oh. Okay. Now that I have my file open, I can just type, this is my new file. Control D on your computer will hit enter, and then Control D will close that out. So just a new text file opened with just some dummy text in it. Once everybody's created just a dummy file that they're happy to send across over to Bill, let me know, and we can do a file transfer. If we're good to just move a file, I'll hop back over to the presentation and show what the next step there is. We're going to run a simple SCP command. For me, I name my file myfile.txt. So whatever file you created, you run SCP myfile.txt or whatever file you chose, your username at upload.brainimagellibrary.org, and then that directory we created before. So for me, I would just do, since I have that file available right here, my new file, I'm going to do SCP, my new file, my username at upload.brainimagellibrary.org, colon, and then that not that. And then I'll put that directory that I created before here. Mine is under bill/lz/my username. And then I'm going to jump back and see what my unique identifier was for that. Stay character there. And if you hit enter, it'll prompt you for your password for Bill. And I successfully just uploaded that to my Bill directory. Has everybody gotten to the point of moving that, or does anybody need any assistance? We're getting some thumbs up there. I'm going to go back to sharing my Chrome screen here. So when I had folks logging in earlier to On Demand, we're actually going to jump back over to that. What's great about On Demand at BIL is it is an easy, accessible area to run things like Jupyter Labs, RStudio. But what it's also good for is you can actually take a look at the file system itself while you're on here and take a look at files you've uploaded or directories you've created. To do that, if you go up to Files and then Home Directory, this actually takes you onto your home directory on the BIL VM. If we want to take a look at that file we just uploaded, we can click Change Directory. And you'll just fill in here with your directory you created through the portal. So mine is BIL LZ, mine username, and this. And if I click OK here, I can see that file that I just uploaded through SCP. Within this interface too, you can do things like view the files you uploaded, edit them, anything you need to do, move things around. Which is nice because when you upload multiple directories, you can kind of do your work here instead of on your local machine if you'd like to. So I'm going to pop back over to the presentation. And that's just what I show right there, the on-demand. If you'd like to reference this again to get back to there in the future, you can do so. You can also access these files through the command line if you like. This is done by, within your command line, you can run an SSH command, which will route you into the BIL VM. To get to the BIL VM to look at your things through the terminal, you would go, oh, I'm sorry, I'm not sharing the correct screen. You would do a command like SSH, your username at, and in this example, you would do login.brainimagelibrary.org. That would prompt you for your password. And just like you saw on the on-demand screen, just takes you right into your home directory on the VM. And I can do a CD into that directory I created before. And I can look at that file here too. So either way, it's just dependent on what you're more comfortable doing and what's easier for your workflow. With that, I'm going to turn it over to Mariah to talk about metadata, as that would be the next step you need to do to finalize your submission.
All right. Thanks, Luke. Katie, I think you had your hand raised. I thought I saw a thumbs up. Let me know
Yeah.
if you need anything.
Thanks. I did. Well, I put it back down, but
Okay.
yeah, I was just wondering, so is there any way I can, like, if I have files in a landing zone and I want to, like, show them to one of our scientists to be like, hey, does this look right? Is there a way I can do that without them logging into my account? Or it sounds like maybe we should have an organizational account or something.
You want me to take that, Mariah?
Oh yeah, you or Luke.
Sure.
Not me.
Yeah, so there's a couple things that can be done. So if it's a user on the system, there's a way that you can set up an access control list to give them access to your landing zone.
Okay.
Part of what I alluded to with access giving us better management of project space is that through access, it will give us the capability to do exactly what you're suggesting. So there would be a common project space for everyone who's associated with your PI or whoever, let's say. So if you have a PI and there's like 4 people under him, all of those folks would be in a common group. We do that now with project space, but that's manually set up. But once we have the access allocations, that'll be automatically set up, and it's simple file ownership. Just changing the permissions on that.
Okay, that's great. Because like, yeah, I'd like to be able to set up like read only access for somebody so I can, let them look at everything without the risk of them accidentally like deleting something,
Okay, yeah, so that in that case, probably the access control list would be the better way to go. Have you used those before?
Not, I haven't like,
Okay, we'll follow up with you and
Okay.
and give you some instructions on that.
Thanks.
And all right, I think can you all see my slides?
Yes.
Okay, thank you. So I'm going to talk a little bit right now about file organization. Before we get into metadata and before we take a break, we'll just talk a little bit about how do I organize these files, what makes sense for how we organize our metadata, and how to structure all of these. So it's right, just a reminder where we are in the submission process. You've already created your submission. You can't upload metadata and upload data simultaneously because obviously uploading sometimes hundreds of terabytes of data can take a long time. So it's important to be able to do that simultaneously, but we're going to talk about how you structure the data that gets into your landing zone. So before we talk about how to structure that, we should kind of talk about a few definitions between what is a submission and what's a data set. Up until this point, we've been talking about submissions, really. The landing zone is for a single submission. And so your submission is going to contain multiple data sets. A data set is really a standalone entry of either a volume, an image volume, or an image set that is from a single subject, so a single mouse brain or a single experimental unit, depending on the different types of assays that you are doing in the lab. So this is going to be a unique metadata entry, usually associated with one donor or a single subject. Depending on how you set up your experiments, it could be a single part of the brain. You're going to have many, again, a single submission is many data sets, and it can typically contain many 2D images are assembled to form a more complex 2 or 3 dimensional volume. So the submissions will inherit the project metadata that we talked about at the beginning. So all of your submission, everything inside a single submission has the same project metadata, the same grant number, the same laboratory name, and the same NIH project. And so everything that you submit in a submission should come from that same project. If you are looking to include, if you have data sets that are from different projects, these should be different submissions. In general, so we said at the beginning that smaller submissions are recommended just because of the way that if something gets tripped up in validation, then it won't hold up all of your data. And if you have different levels of data, so you have some processed data, you have some raw data, these should be separate submissions so that the metadata can be clearly assigned to each of your data sets. So big takeaway from that is that a single submission is going to contain multiple data sets, and each data set represents a single donor or individual or an experimental unit, depending on how you set things up. So if we look at this example. So a single submission branches off into these two different data sets. You could think of this as a whole brain. And so each of these small boxes here is a different subdirectory within your submission. So data set one would be its own subdirectory within that data set, which in this, if you're looking at, maybe a whole brain imaging data set, that would be a single mouse, and then you'd have the different channels for the data set would be their own subdirectories, and then you'd have the Z-stacks within that. So you can kind of flow, you can see here how the data would flow through the different subdirectories and how you would structure that. Let's talk a little bit about some of the best practices whenever we're structuring our data. We do suggest that you do a general file cleanup after your transfer. Luke showed how to do that in on-demand, just to check that the file sizes make sense. Did everything finish uploading the way that it should? Does everything make sense for what you uploaded? Is the organization what you thought it was? And make sure that everything uploaded without any errors. Also consider file format. It is you're sharing your data, and you want it to be accessible to the larger scientific community. So try to avoid any proprietary formats or anything that's not going to be accessible to the community, anything that needs a special software to upload or to open. Whenever you're uploading your files, do avoid any special characters. So anything that's not an underscore, a dash, a period, or numbers or letters, including any spaces, can introduce errors, and we will change that in the validation process. Whenever you're uploading your submissions, make sure that you can separate each of the data sets or specific donors or individuals into a unique directory. We're going to get into the metadata file after the break. And so you'll see that each data set needs to have a unique path. And so whenever we list the metadata in the file, the metadata file, you're going to list all of the directories for each of the individuals. And so each of these should be unique and should not be repeated. And finally, if you have any extra metadata files or something that you'd like to include in your data set that are not any image files, then you can create an additional subdirectory at the root of your submission that includes any of your extras, so that all of that metadata can be included, but is it structured in a way that makes it possible for us to visualize your data down the road. Any questions about file organization before we take our break? I realize it's kind of a high level right now, and we will talk about it more when we talk about the metadata file. And I do realize that it is hard to give this advice whenever there are so many different modalities. So, and your image data might not be structured like the way that a whole brain imaging is. So we do have different options for this. This is just kind of a high level recommendations of how to structure your data. Of course, whenever you submit your data for validation, I'll be looking through this and give you some suggestions, or if you need to upload your data and then reorganize it after it's on the BIL system, that's definitely an option. So we can talk about all of those whenever the validation process has begun. But any questions there? Yeah, Katie.
Hi. So I want to apologize for uploading 300 terabytes of proprietary image format data.
While we're here, it's okay.
I was asked to do it, and yeah, well, it's like the alternative is convert. We have process output data, right? So I guess I had a couple of questions, and like one, you said, so process data should be separate, a separate submission from
Mhm.
raw data.
That is true, yes.
Why is that?
So the reason is because whenever we think about the reuse of the data, you want to be able to quantify what, or really not quantify, sorry. You want to be able to specify what data was actually used and cite that. So we're looking to really go down to what is the individual citable, reusable piece of data and index it at that level so that it's clear for reuse that if someone goes and reuses your data and they didn't reuse your raw data, they used your process data so that they can cite that process data specifically.
Oh, okay, I see. That makes sense. Yeah, because we had a scientist I was working on with wanted to put the process data and the raw data like in sibling folders,
I see,
like
yeah.
in the same section. So because they felt like that way it was easier to link up which like the raw data, which is proprietary format mostly,
Okay.
unfortunately, and then the process data, which is like TIFF files, etc., like some cell by gene matrix, that kind of thing.
Mhm.
And that's in the folder like right next to it, so it doesn't. I'm
Yeah.
just wondering.
Yeah, so I mean, so the definition of raw data is a little fuzzy because we are not looking for your proprietary files for you to share unless you've truly written that into your, unless you've written it into and it's like an agreement that you have with the NIH that you have to share these proprietary file formats. Then, it's not great for the scientific community though. They don't want to, it's not as accessible. Right. And so raw data is, yes, it's the raw level of data, but in a format that's accessible.
Okay. Yeah, we'll have to.
But
We'll
if
have
you
to
do-
talk more about that, because I think it was like we did say, okay, we're going to share everything, and so like we kind of have to do it, but we kind of realized afterwards that it wasn't.
Yeah, I
like,
understand
super
that.
helpful.
But the good thing is that, if you do need to compute on your data to transform it or to do any, to do any transformations on your data, you can do that on the BIL cluster after you've already uploaded it. So you don't have to upload again, or any of that. So, yeah.
Yeah,
Oh,
but
yeah.
I just want to add one more thing to what Mariah says. And I mean, one of the reasons why we have a more flexible ingestion process is we realize that we can't handle every condition that's out there. And sometimes the technology rapidly advances and there is a great benefit to the scientific community to making the rawest form of the data available. For example, if people are developing new ways of processing the data, there is some benefit to making that available as well. So in general, we're kind of flexible. Mariah's guidelines are excellent, but we do realize that there's always exceptions to the best practices that she mentions.
Okay, thank you.
Does anyone else have any questions, concerns? We can take a break and come back and talk about metadata next.
So what time did you want people back by, Mariah?
What is it now? It's about
It's about 2:10 Eastern.
10. So yeah, let's take 15.
I'm wondering, is there more time for like other questions? I was kind of holding my questions earlier, but like, or maybe at the end, or I don't know when would be good.
If it's questions about, what we talked about in the first half, I think it's definitely good now.
Yeah, just some stuff you talked about in the intro. I was trying to like not interrupt too much, but I was just wondering, so is there like redundancy in your storage systems, like error correction, that kind of thing?
redundancy in the storage, does
Like,
Luke? Or
yeah, so,
would you like to
like
take
for
this one?
example, like with like, you know, like if you upload your data to like Amazon Web Services, like there's some kind of like they check the hashes periodically, and then like if there's like a discrepancy, it like corrects it. I'm just wondering, is there redundancy?
So I can try to handle that.
Yeah.
I mean, there's always the trade-off here between error, redundancy and backup, right? So
Yeah.
there's an effort to make a tape backup of all the data that reside on BIL, and so there'll be a direct backup on cold tape medium. But then there's also all of our file systems are backed by ZFS, and so they routinely do a scrub, right, where you're talking about the checksums. So block by block, everything will be checksummed and auto corrected if bit errors are detected. Does that answer the question?
Yeah, that does. Thank you. And then, so what is involved in the data validation steps? Like, does that like?
We are going to talk about that more in the second.
Oh,
half.
okay. Okay, good.
We'll,
Then
we'll talk
I'll
more about
save that.
validation.
And then I was just wondering, so the rsync transfer message, it looks like that's also sending stuff over SSH. Is that correct?
I believe so.
rsync.
Yeah.
Yeah.
Sorry about that.
rsync is also over SSH. Okay. Does anyone know how much a Globus standard subscription costs? It doesn't say on the website.
So it depends on what, you know, it's like most software that's out there. It really depends on your institution and
Okay.
how much they think your institution has to give them. So
Okay.
I can't give you a good number, but I know that, you know, for a large
Okay.
research university, it's I would consider it very expensive and more than what, you know,
Okay.
more than what a cheap car would cost new.
Okay. Thanks. Okay, that's all the other questions I had. Thanks for taking them.
All right, so let's take 15. So we'll meet back at what is that, 27?
So why don't we just make it 2:30? We'll be back.
You wanna be
And,
back at
2:30 2:30 Eastern, yeah. We can give everyone an extra minute or two.
All right, thanks, everyone. All right, hi, everyone. Get ready to start back up again. Let's get all of my screens set up here. All right. So the next thing that we're going to talk about is the metadata file and all the information that you submit along with your data to make sure that it is accessible and easily understandable to the scientific community. And so we've gone through creating an account, requesting access, creating your submission. The first part of this workshop was really focused on uploading your data, getting it to where you physically need to move it to. But now let's talk a little bit about metadata. So the goal here is to really increase the fairness of the data that we have in BIL, make it accessible, reusable, and the metadata schema that we currently used that is designed specifically with DOIs in mind, and the ability to issue those to make sure that we're capturing all of the information needed there. We are transitioning to issuing DOIs for data sets immediately in the near future instead of by request. And we will talk about DOIs at the end of this presentation, or at the end of the workshop, sorry. The implementation of this is in a spreadsheet format. So it's easy for you to include all of the information, your metadata of the samples that you have. And you'll see that we have some metadata models in place that are ideally there to reduce the amount of redundant fields that you have to include in your metadata, and to make sure that we can easily link data sets from the same sample or the same project. If you would like to read more about the metadata schema, the preprint is available there. But just a quick overview, and we're gonna really get into the weeds on what's included in this file. But of course, you have contributors information of who contributed to these data sets and what were their roles, funding information. You have the ability to include any publications, preprints, protocols that are associated with the data sets, information on how it was, the images were captured and the instrument used, data set information with abstracts and how, and the directories of where to find that data, specimen information on the samples, and of course, image data and anything needed for to describe the images and their orientations and resolutions and things like that. So whenever you choose your, you're going to choose a metadata model whenever you upload your metadata. These metadata models describe the structure of your submission and how the data sets are related to each other. Choosing the correct metadata model is important because it makes sure that your data is properly ingested and also indexed within the database so that we can properly link things that are related. The metadata won't be able to be uploaded correctly if the metadata model isn't selected and the structure of this metadata file isn't correct. I think we have someone in the waiting room. There we go. And there are 4 different metadata models, and I'm going to go through each of those here. So whenever you go to the Brain Image Library Submit portal, you are going to be asked, what does your data look like? And it describes all of these 4 different metadata models. We're going to talk about each of them, but this is also where you can download the metadata sheet. So let's do that right now. Let's take a look at the Submit portal. And if you go to, one second, waiting room. So if you go to the Submit portal, it might look a little bit different than yours. You go to New and then Add Metadata. You'll have the option to download this metadata sheet that we're going to be talking about right here. You can download it in Excel. I'll download it there. But let's talk a little bit. So you can open it up and go through this and see what it's going to look like while I'm talking about this. But let's talk a little bit about what each of these models are before I go into all of those details. So if we look at the first metadata model, it describes it as that's the first option right here. Each data set was generated from a single sample or donor. So each data set that we have, each unique directory is from a different sample donor. Possibly this is most likely a mouse, or if you have human data sets, an individual from the same instrument. So multiple data sets can be accommodated in a single submission. The lines on each of the specimen tab in the metadata sheet are going to exactly match the lines in the data set tab. So the specimen that's listed on row 5 is also the specimen that's listed on row 5 in data set. I'll explain that and show that to you. And each data set tab would have a corresponding image tab. So this is a little schematic that you'll have the instrument tab. There's going to be one entry, and then the image tab, the data set tab, and the sample tab are going to have 3 entries each. So for metadata model 1, so we're talking about these different tabs down here. The first 2 tabs are contributor information. We're going to go through all of these more in detail, but whenever I'm talking about rows, this is what I'm talking about. This row would have 1, and then there will be multiple on data set, specimen, and image, just so that that's clear. We're going to go through examples of all of these. This is just an introduction. So for the second metadata model is that each data set was generated from multiple samples, specimens, or donors on the same instrument. So this is only a single data set can be accommodated per submission. Generally, this is more of like a process data where you cannot separate the specimens in the different into different images and different into different directories. So there's multiple specimen information in a single file. So there should only be 1 line on the image tab and 1 on the instrument tab. So there's only a single data set, but there's multiple samples included in this. For metadata model 3, all the data sets are generated from 1 specimen, but they're from unique regions of interest on the specimen. Multiple data sets can be accommodated in a single submission. The specimens will be listed multiple times in the specimen tab, though. And the region of interest is identified in the locations column. So we'll see what that looks like. Each tab will have a corresponding image tab, and there will be exactly 1 line on the instrument tab. So we have 1 sample listed. There are multiple data sets coming off of that, multiple images, but only 1 line on the instrument tab. And then the final metadata model is that all data sets are generated from a single specimen, but use different experimental parameters. That means multiple data sets can be accommodated in a single submission. So we have multiple data sets, a single sample, there's one entry on the specimen tab that's shown here. The lines that are listed in the instrument tab match exactly to the lines on the dataset tab. So dataset, image, and instrument are all corresponding. So let's look at a quick example, and then we're going to go through the whole file and talk about all of the different entries in there. So if I were to have 8 image files from 4 different samples of different mice from the same part of the brain using the same microscope image settings. So for this, I would describe that using metadata model 1. So I have a single sample or donor for each instrument, but there's multiple data sets. So if I were to organize these, I have my 4 different mice on the left here, and I have multiple TIFF images for those. I would structure those in a way that I have the BIL, landing zone, my user ID. Then in a single submission, I have these 4 different data sets. So let's look about how we would fill that out in the spreadsheet. So when you, let me make this full screen. When you open your metadata file, sorry, I'm seeing that we have a question about where these are all located. I should have maybe included that previously. Let's go to Google. If you go to the portal that was included in the email that was sent to you all, I can also, can someone drop a link to this in the chat as well? You can look at the GitHub. All the presentations and the scripts for both days are listed in the GitHub here. The presentations are all in this directory. If we go back, there's also metadata models. So these are examples. These are the examples that I'm going to be going through. So if you want to look at the files that I'm working with, they are all included here for you. All right. Let's go back to the presentation. Let's look at, this should be the same file that either you, if you want to look at it on the GitHub, to download it from GitHub. If you want to download it from the submit portal, it's going to be the same file. And so you can look at the overall structure there. First tab is a README that includes descriptions of what's included, complete all of the tabs. There are things that are required. There are things that are controlled vocabularies, and there are examples included in these. So let's get right into it. For the contributors file, first you start with who contributed to this data set, who's responsible for it, and who should be listed as really an author, or author as it were, if listed on the DOI landing page, and who should be included there. So if I were to fill this out, my name is Mariah Kenny. So I'm going to put Kenny space Mariah. Creators are the main people who are involved in this, and they are indexed in the, they're going to be listed at the very beginning. So you can have people who contributed, but you also have people who can create the data set. So if you want someone to be listed within the author list kind of the data set, then they should be listed as a creator. But if you just want to recognize someone as someone who contributed to the data set, you would say no here. But I'm going to say yes, that I was a creator and I contributed to making this data set. So you have different types of contributors. You can have different roles. I'm going to put data curator because that is what I am. But you do have other options here for the controlled values are listed. You can either be a contact person, project member, project leader. And we ask that you only include a person a single time and select the most appropriate role for them if they have multiple roles. Organization type or name type. So this can either be personal or organizational. If you'd like to list an organization as one of the contributors for a data set. But for me, I am a person, so I'm going to put personal. Next is your ORCID ID. We do, or name identifier. For most people, this is going to be ORCID ID. If it's an organization, you might, you won't include this here. If so, I don't know my ORCID ID off of the top of my head. I could look it up, I suppose. But let's do that. Why not? So I'm going to Google myself. If you don't know your ORCID ID. There it is. So this is my ORCID ID. And that's exactly what you will put in the name identifier space. And there are other options such as RID, SNI, ROR, there are different options there, but I'm using ORCID. That's probably what most of you will use for anything that's in personal. My affiliation, I'm going to copy and paste from here because I know that I work at Pittsburgh Supercomputing Center, but if you're unsure of your organization, you can always go to roar.org and search your organization. So if you wanted to, my search history is strange here, but let's say I'm at the University of Pittsburgh. And so you can see here, this is the ROR link for the University of Pittsburgh. So I put that included here. So if you ever need to look up your ROR organization, you list that. And then I'm going to put ROR because that is the scheme of the identifier that I've included. Please let me know if there are any questions at any point. So for funders, you can list multiple grants and funding organizations here. So if you were to have, if you know your grant award, but let's say maybe you need to look up some other information about it, you can go to reporter.nih.gov. This is the full, even if you only know part of it, you can search for your grant here, and this is the grant number. You'll copy and paste that into the award number. I know this is NIH, so I'm going to put NIH here. This is the ROR ID for the NIH. If I were to go there, that's the ROR ID for the NIH, which is the funder organization here. So I'm going to put that there. This is ROR, and then the award title that you would get, from Reporter. I think I closed that already, but I would put in the title of the award that you have, and that goes on that line. For publication, you have a lot of options here. It's a bit flexible. I encourage you all to include whatever protocols, preprints, or publications that are associated with it, with your data sets. This really does increase the accessibility of them and make sure that people are citing and accessing the right data sets that they think are associated with a specific manuscript. So you can put a DOI here or a PubMed ID. You have a few different options here in the relation type. For the most part, you'll probably be using is cited by or is documented by. And then the citation in whatever format you'd like to include for the resource that you are citing.
I have a question about this particular tab. Are people generally uploading this data that's presumably in that publication after the paper is published, or how does that work?
Yeah, so if you know what the DOI is, you know, for your preprint or for your protocol before you're publishing, before you're submitting your data, that's great. But if you don't know it, it's all right. We can add it after the fact, but you can still add things. But I understand that you might not know that before you are submitting. Does that answer your question?
I guess after the
You would have to reach out to BIL support in order to add it. That is accurate.
I guess if you're uploading after the manuscript is published, that's actually the one I'm asking about. Like, how do people normally manage that? Or
You're uploading your data after your manuscript is published.
like, say you uploaded the preprint,
Mhm.
then you have the DOI.
Mhm.
Is there
Yeah,
any?
and you could include that DOI here.
I see. And
But
then
if you have a DOI for, then you wouldn't have your DOI for your article until it was published later, then we'd have to add that afterwards.
yeah, but the published article wouldn't have the links to that data.
Yeah, so ideally, you would start uploading your data and you don't, doing those two simultaneously or getting your data out before your publication is even ready. These two the publication and your data being published don't have to necessarily happen at the exact same time. It is ideal to kind of have your data come out a little bit before your publication so that if your journal is asking you, I need a DOI for your data set before we will publish your article. That happens sometimes, right? So do try to kind of plan ahead there and make sure that you can get your data set out and through the validation submission process before your article is ready to be published.
I see. Okay, thanks.
Yeah, and if you're talking about sort of a legacy publication like that, maybe you published several years ago, and you're hanging on to the data and you want to make the data publicly accessible and submit it to BIL, you certainly can do that as well.
Okay. Okay, thanks.
Mm-hmm. All right. So those first three tabs of this file are going to be the same, no matter what model you use. But the first one that starts where the four that start to differ depending on the model are instrument, data set, specimen, and image. So just to remind you that we're looking at where we have 4 different samples. I have 8 images from 4 different mice. They were all using the same microscope. So if I only have one microscope, I would list my microscope type here. Maybe it's light sheet. I would list the model of the microscope that I have. These are 2 just this is these are both open text sheet or entries, so you can put whatever is appropriate for you there. These 2 are in red, so these are required fields in this metadata, and then the rest of them are optional, but I do encourage you all to provide as much information as you can about your data sets. So some of these will be applicable to you, some of them maybe they won't be. And so please do fill out what you can: objective name, immersion, numerical aperture, detection type, illumination, wavelength, things like that, and the temperature. All of these are going to be important for data reuse, anyone who's trying to use your data after you've published it. Yeah, Katie?
Yeah, I have a question about instrumentation. I think I remember on some of your earlier slides showing, if you have multiple, like you have multiple subjects in one submission.
Mm-hmm.
what if you have like multiple instruments being used on the same like subject?
Multiple instruments used on the same subject.
Yeah, like
Yeah.
there's several devices involved in generating the data, but the way that at least I don't know if there's a format for that where it's like you have multiple subjects, they're all using the same series of instruments, but I don't know if that's like
Multiple instruments.
yeah, it's not multiple instruments. It's not a different instrument for every subject. It's just like the same instrument or the same subjects are all, have the same process done on them, but there's
Protocol,
multiple
yeah.
instruments involved in the process.
So, I mean, let's see, we might have to talk about that specific case. If you want to list multiple instruments, but it's kind of, they're all applicable to the same one, because right now it is set up in a way that you would list your instrument just on one field. I think it depends on kind of your protocol and what's coming out of those. What your data really looks like and how we could break it down. Because everything is listed in a way that it would be one row, and so we'd have to really look at that and see how to interpret that for your specific data set and see, does that need to be broken up into multiple submissions, or is there a way that we can kind of represent that in this in these fields in a way that kind of doesn't break any of the models that we currently have, or maybe we need to kind of.
Yeah, I'm just like trying to so just for example, and I don't know all the details of this, but just like looking at illumination and detector wavelength, right? Like if you have spatial transcriptomic data that has multiple wavelengths, you're going to need multiple entries on this sheet in order to like capture all those wavelengths.
I see. Well, so I will say that normally that these people do include a range, but I see what you mean there. I think for spatial transcripts, we might have to talk about that one specifically of what your data looks like.
Yeah. I'm not sure all the details, but it's just like, that's just the first thing I see there is that like, if you want to list multiple wavelengths, I guess you can just give a range. I mean, it's probably fine. But yeah.
Yeah, these fields are, these are open text fields. You can kind of put whatever information you have in these fields, and whenever you submit it, it will be curated. And if I have any questions or if it's not clear, then we can change it. What you submit does not have to be written in stone. It can always be changed and altered depending on after, I see the data, and we can decide what kind of makes sense moving forward.
Okay, great. Thank you.
So let's move on to the data set tab. So for these four different, I have these four different samples, 8 images, decided that these are going to be 4 different data sets as we've described. So for the directory, you will put in the BIL, LZ, and then your username and the submission ID. And then for each of these, I'm going to have a different data set for each individual that I have. So this will be mouse 1. And then I will have this for, be very careful with what I'm doing. I'm doing this because I have mouse 1, 2, 3, and 4. This drag and drop feature in Excel can get you into some errors if you use it, not knowing that it's not copying and pasting. But I have mouse 1, 2, 3, and 4 because I know that that's how I'm going to structure my data sets in the landing zone. For each of these data sets, they're all going to have a title. So mouse, I'm going to just put mouse images. And then this is for mouse 1. And then same thing here, but this is for mouse 2, because each title should be distinct and unique depending on what the data set is. I'm going to do that for each of those. So it's the same title, but clarifying that it's for each different data set. If you have anything for a social media post, some text or anything like that, there's some subject fields that you can include, key phrases, classification codes, subject scheme or the scheme or the classification used for each data set. These are all optional fields. Then if you have rights, so what is the licensing information that will apply to your data set? So CC BY 4.0 is what I've chosen here, and then list the URL for those. So in most cases, you'll probably be using a Creative Commons license. There are many options for that, and I believe all BIC, not BICCN, BICAN data now is all CC by 4.0. If you have an image or a landing page, or a web landing page for a data set that you'd like to include, that can be included here. 2 more required fields are the modality. These are controlled values of what you can include this modality. If you're BICAN, it should match what the modality is that you use for BICAN, along with the technique. So I've got connectivity and interrogate tracing. There's many other options here. If you've selected other, then you can use this field to describe either the modality or the technique that you've included that's not listed here. And then an abstract, this should be at least a few sentences long, describing the data set and what's included. And then any methods. So ideally, if you have a UI for a protocol or any other notes about the method that was used to collect the data sets, or any technical information about is there anything needed special to view these images, or was anything any computational information needed to really understand what's happened to this data before it was published. So we have 1 row here for each of the data sets. Next, if we go, because we're working with model 1, we have the specimen tab. So the first column is local ID. So if you have an ID that was used in the lab, anything that is unique enough that you will be able to kind of tell what individual you're referencing. Things like dataset 1, dataset 2, those aren't as truly useful for us. We want to be able to make sure that if you're including data sets that are across multiple submissions, we want to be able to link those. And so including ID here that is unique is really important. And so if I have some unique ID, some number that makes sense for each of my data sets, and they're all unique. That species minus mouse, I'm going to put that here for all of them. NCBI, taxonomy. If you need to look that up there, it's the code for each of the species. Then I have some of these pre-populated just so that we can get moving a little, not spend too much time on it, but age, age unit, and sex of the of the organism, to include information like genotype here, a local ID for the organ or tissue, for the organ listed here. Most of your data sets will probably be brain, but there are some spinal cord or data sets in BIL. So you do have that option. Then a sample local ID. So if I was using, the same donor, then and I took multiple samples from that donor, this would be important for me to include. But if not, that's okay. Then if there was any atlas they used and in locations of where that tissue is found, you include that on the specimen tab. And then the last tab that we have for this example is the image tab. So you list information about the orientation, any oblique information with these, this axis, the X, Y, and Z axis, all of the predominant tissue directions for each image that you have, any landmark information, the channels that are number assigned to each channel, and the display information for those. And then some of these might be applicable to what you have, and some of them might not be. That's why they are optional, but you do have step information. Information about pixel size is really important to include here. And then channels, slices, time points, total size in gigabytes files, dimension order, anything that's applicable for you, please do fill out as much as you can. And so that is the end of our first example here. Let's go back to presenter mode. Any questions about this file at all, or this example that we just went over?
I think I know the answer to this, but the blue example text that you have above the where you provide examples of what types of things to put in, that can remain, right? We don't have to delete that.
Yes, do not delete it.
Anything below that.
That's a great question. So everything that is on, line 7 here, keep the example information. Everything will be read starting at line 7. And so it's important that, you're lining up the first data set is on line 7 of the data set tab. Then it's also on line 7 of the specimen tab, and it's also on line 7 of the image tab, right? So all of those line up, and that's important to kind of keep track of.
Got it.
Right, thanks. All right. So for the next example, we'll go through a few quick examples of the rest of the metadata models. So if you were to have image sets taken of 2 different magnifications of the same mouse, for that, the metadata model would be 4. So we have all the data sets are generated from one specimen, but they use different experimental machinery parameters. And so, if I have 3 different images related to each of these different magnifications, I would structure them like this. And then if we want to take a look at what model 4 would look like in a metadata file, as I said before, these will still remain the same. We'll have 2 different instrument tabs here, 2 different data set tabs, a single specimen tab because, or a single row on the specimen tab because it is the same specimen. And then because these are 2 different image sets, they will have 2 different image set metadata fields or metadata entries. So if we look at, so we have 4 different image sets for 4 different regions of interest of the same brain. And we will structure our data set like this. That metadata model would be 2, so that the data sets were generated from multiple samples on the same instrument, and only a single data set can be accommodated per submission. Multiple specimens would be listed in the metadata sheet, but only on a single data set tab. And then quickly, if you were looking at a case where multiple samples were included with unique metadata, but the samples couldn't be separated into different files or directories, that means that there were multiple samples included in an image or a file for more processed data. So that would all be included in a single data set. And so therefore, that's how you would structure your data set there. So it's harder to kind of to tease out the specimens that are included in a single data set if they're all included in the same file. So that would be the last metadata model that that there is an option for. And if you have more modality specific questions about what model should I choose to reflect my data, and how do I kind of structure this file, we do have office hours that meet virtually each month for hands-on assistance. They are scheduled for the second Wednesday of every month at 2 to 3 Eastern. There is a link here to register for this. You can add this to your calendar, and then once you fill this out, you'll be sent an invitation, and it'll it's for the Zoom link and all of that. So if you do want to come, and Luke and I are both there at these office hours, if you ever have any questions on how do I structure my data, how do I fill out my metadata file, or you just have other questions about the process, you can find us there every second Wednesday of the month. But also, if you can't make this, that's okay too. We are glad to set up a time to speak with you, or if you want to just have questions, you can always email billsupport@psc.edu. And we're happy to work with you to figure out how to make sure that you can make your data public and publish it and get it out there for the scientific community. Any questions on what I went over? There are examples, but if any of it's not clear, please do reach out to me and I'd be glad to talk to you about it. Great. Thank you so much. I think Luke's going to talk about how to finish this. How do you upload it and submit it and make it public? Let me stop sharing.
Mariah, would you be able to share the filled out metadata file with folks in the chat to download for the next exercise? The GitHub link to that.
GitHub link to that. It is in the portal, but I will go and specifically go to a metadata models. Yeah.
Just so everybody has the same document we're going to be working with here.
Yeah, you can use any of those, but there you go.
Yeah, just grab one of those for the next section. We'll just be uploading one of those, and just so everybody gets to see what it looks like after you've done a metadata upload. But we'll move on here to what Mariah was talking about a bit. Mariah showed this page earlier where you guys went and downloaded a blank template for the metadata. This is also the page that we'll be using to upload that metadata associated with the submission we created earlier today. So if we go to that page on the submission site, if we go to new and add metadata, depending on the metadata sheet that you grabbed, and I'll do the same. For example, I'll grab the model 1 here and download that. Since I have the model 1 metadata sheet that Mariah gave here, I'm going to choose option 1. Scroll down to that associated collection that I made earlier, which was my April BIL workshop collection, or submission rather. Sorry about that. Upload metadata. And I'll choose that document that I just downloaded. If I can find that. Give me 1 second here. And I'll click open on that. We'll get prompted that the portal itself is processing the metadata sheet. This can take a few seconds. And now it kind of gives you a view of all of the metadata that you uploaded with these data sets. For example, this is shown in a data set form. So each one of these data sets, if you click on view more, gives you more of a detailed view of all the metadata that you gave. Just to clarify with folks too, up top is this may cause some confusion. Up top says metadata version 1. About a year and a half ago we switched models into this newer metadata schema that better suits DOI creation. And as a result, we've kept legacy data around just in case people want to access things that were around before we moved to this new model. But when moving forward with submissions now, this is the section you'll see information get populated in once you do that. So I'm going to jump back over to my slideshow here. So now we're at the point of we've uploaded data, we've uploaded metadata, and we're ready to make the data public. To let us know that you're finished with both uploading data and metadata, and you've looked over things and you're happy with the way things are formatted. On the submission portal, there's a button called Submit Validation Request. What this does is it will send us an email directly letting us know that this user has requested this data submission be put through the validation process and a public link to be generated. If I go onto the submission portal, up top, this submit publication request, and I click that. If I scroll down to my submission that I did earlier and click that, you can also choose several of these. So if you have several submissions, you're doing it once in one big batch, and you want to let us know that, several of them are ready to go, you can click multiple. Once I've chosen that one and I come down here to hit submit validation request, it says request successfully sent. So that will get emailed to us and we'll be pinged to put that through the validation process. Oops, I lost my tab here. And we will get back to you notifying you when the validation's finished and a public link has been generated for your data. So what happens during that validation period? We run the data sets that you submit through several different sanitation checks. We check to see if there are any empty files through the many files in the directories, if there are any naming schemes within the file names or directory names that would trip up a file system. For example, if there's a space, we'd run this through and just replace it with an underscore, et cetera, things like that. But the longest portion of it by far is certainly when we run it through our bioformats. We run it through bioformats just to make sure the images are usable. Essentially, this checks and sees if they can A, open the file, or B, if the file that they open is actually something that has an image there, or if it's something like a black screen or just there's nothing there, that would kick back and let us know, hey, there might be something that happened where a file got corrupted during transfer or something like that. So we get that report back from bioformats, and then finally we curate the metadata and confirm that it's the same as the metadata submitted, and then we run a move on everything. So the validation says the validation of data does not necessarily mean the quality of it. It just is if it can be used or if it is having any errors or things like that. It's dependent on the size of the files, like Alex said, like the size of the files of the amount of files. And we already did this here. Sorry, I think I jumped ahead of myself. This is just doing that, sending a request to us, letting us know that your data is ready to be public. At this point, the data moves past validation and gets a public link generated for it that is public facing. Just to reiterate what Alex mentioned prior, the amount of files and the size of the files is what will cause a longer time in validation. Just keep the rule of thumb. If it takes a while to upload, it likely will take a while to validate as well. Lastly, what do I do after my data is public? How do I get to it? There's several different means of getting to your data. We provide HTTPS links to all the public data at BIL on the download.brainimageslibrary.org website. The link for data will always go as following. If you made note of when you made a submission, the unique identifier at the end of it is just a long string of characters that remains unique to your submission. But to get to that after it's public, we always structure it as the same. It would be / the first two characters of that UUID / the second two characters, or sorry, / the third and fourth character, and lastly, the full unique identifier. We do provide the HTTPS link as well as this data is accessible either via on demand or through the command line itself. If you were to log in and look at things that way. You can take a look at all the public data at BIL on the submit.brainimageslibrary.org/search site. I'll show that here. The data search tool is it's a queryable interface to get a look at all of the public facing data submissions located on BIL. And they provide links to the data, both on the HTTPS links and the actual directory it's located. I'll show that here quickly just to get an idea of what you can do with that search tool. So it's just the same address as the submission site, but then a /search at the end. These are all the public facing data sets we have. For example, if I wanted to take a look at this this spatial transcriptomic data set, I could go either through the public link and pull things that way, or I'll give an example with on demand. If I wanted to look at it on demand or through my command line, I can just copy the directory here, look at things directly that way. From here, I can take a look at things through this interface, or if I log in, I could even open some of these things in Fiji or run any sort of software we have available on them to do your own kind of experiments with things. I mentioned early on in my presentation about the BIL PI / data manager dashboard. Essentially, we have additional features on the submission portal available to data managers and PIs. We can give these additional features if a PI or a data manager just sends an email to BIL support letting us know, hey, I've been given the okay from my PI to look over as the PI or data manager of our project on BIL. Essentially, what this lets you do is, if you are a regular user, you can only kind of see things that you've done. But if you're the data manager or PI, and you have multiple people submitting under the same project, you can get a larger view of everything that people have done in your group. Essentially, you can just manage things and see where everything is. There are also there is also the ability to create more projects and add people on your team to those, so they have another tag to submit data to. We're looking to a future tool as well for a downloadable report with all this info of your project. So you can pull that down either an Excel or CSV or tab delimited style file and have that available to you. Here's a couple more screenshots of some of the extensive detail with the PI dashboard. At the bottom there, you can see there is just a couple project submissions within our project. I can see all of them. I can see the personnel and the several projects that I oversee as the data manager or PI. This isn't required to have somebody on your group as a data manager or PI. It's just additional help for folks to get more information out of what they've submitted at BIL than they normally would. In the future, we're looking to add extra features to the portal, such as a self-service pre-validation. So portions of that validation process that I mentioned, we're looking to allow an automated system that a user can come in and click a button and start those pre-validation steps before they run through the larger ones, just to break down on time between the wait between submitting and getting a public link. We're going to have those things roll out slowly as they come. So keep an eye out for updates with that. We'll send out email when there are updates to the portal, and there will be, announcements on the Brain Image Library website itself. And back to Mariah here on how to cite your data sets. If there are any questions or anything like that, you can also unmute and let me know.
Yeah, I have a question. So does the validation scale linearly with the number of files?
Depends on the file size, individual file size. So
Okay.
yeah, it's really hard to give an estimate of how long X amount of files would take to validate.
Okay, yeah, I was just wondering about scaling.
Yeah, at the same rate, Katie, too. I mean, if it works the same if there's a ton of small files, even. I mean, file systems have a hard time with working with a lot of files as it is.
Yeah.
So I mean, if it's if it's several 100,000 small files, it still will take time to do, even though they're small. It's just a matter of how file systems work, unfortunately.
Yeah.
Yeah, and I mean, it also kind of depends on a lot of other factors too. So in the validation too, it also depends a little bit on the file type that's being validated as well. Bioformats is a really good tool in that it can open up almost any image file type. But some files it can open up quicker and give you that information than others. It's a more extensive process. So it also kind of depends on how that file was structured, and at least it also and the final factor, which really shouldn't necessarily be a factor, but we've noticed this in that when certain projects have data submission deadlines, and it's kind of everybody waits till the last minute to do their homework. So we do get backlogs that we need to manage, and sometimes that slows things down as well.
Okay, thanks.
Yeah, things are curated and validated in the order they are submitted. So
Yeah.
Any other questions before we move on to citing your data sets? All right. The last topic we're going to talk about today: how to cite your data sets. How are you going to share all of these hundreds of terabytes of data that you've uploaded to the Brain Image Library? So a DOI is the golden standard of how to cite your data sets in publications, and I'm sure I'm not telling you this for the first time, but we are transitioning to a DOI system for data sets. And every data set that's in BIL will be issued a landing page that will contain a citation for each data set. If your data set does not have a DOI at the current time, but you do need it right now, please do reach out and we can issue a DOI while we're transitioning into getting all of the data sets in BIL issued with the DOI. But quickly, I want to make a few recommendations of how not to cite your data in BIL. Previously, Luke was talking about, the download link and how this will take you directly to the file system and show you, here are all of the data sets for this specific submission. And while this is great to kind of quickly access things or quickly share things, it doesn't include all the information that you would need to share your information. So we don't suggest really putting that download link in a publication. And also we ask that you don't include a query in your publication, such as all of the data sets in BIL for grant X. We want to make sure that whenever you're citing your data sets, it's a snapshot in time of the data that you were using, and it's very clear of exactly what data you're trying to cite. And queries can change over time, of course. And so it's very important to have a DOI issued of exactly what you'd like to include. Here's an example of a DOI data set. So all of that information that you were asked to put into the Excel sheet earlier, all of that information is displayed on the landing page. So it includes the title, the abstract, all the technical information. At the bottom, it includes contributors, any related identifiers. So if you had a preprint or something like that, it would go here. And then, of course, a link to download the data. Oops. Then something that we haven't talked about yet are group DOIs. So you have all of these data sets, of course, because of NIH, these new policies from the NIH year, most of you are likely required to submit data every quarter. Or so you might have data that you've uploaded over a course of a year even that you want to cite all together for your publication. And so we can issue DOIs for groups and collections of data sets so that they're all easily accessible in one place and you have one DOI to include in your publication, packaged up very nicely, easily accessible. And so we can issue DOIs for this. So it includes multiple data sets here and includes all of the same types of information, technical information, methods, abstract, and things like that. The way that you would do this is by reaching out to BIL support, and we'll ask you to fill out a form that includes all the data sets that you'd like to group and specific information about the publication that you have. So let me show you an example actually of what that looks like. It's very, very similar to the files that you've already filled out. So there's a README, of course, with some basic information. The contributors it's pretty much identical. Includes all of the contributors for, this can be it can be the same list of what's in your publication, but it doesn't necessarily have to be. There are different people who can contribute to the data or contribute to publications. Or they should be listed in different orders and they might have different roles. And so you can list all of that information on the contributors. Of course, you can list your funding information, which is required for NIH projects. Then any publications, preprints that should be included. This is very similar to the previous form that we went over. And then I should include here, this publications tab is also where you will include all of the data sets that you're looking to link. So this example here shows this is the download link for this specific data set. If your data set already has a DOI issued, even you can include the DOI here. If you ever have questions about what to include here, or finding the links for your data sets that you've put it in, or you want to reach out to the help desk and say, I want to include all of the data sets from these 3 submissions. That's fine by me. I can help you create a list of all the data sets that you have and really fine tune that to see exactly what you should include in your data set there. And then description is just a single title, a single abstract, any methods or technical information that need to be included for your group DOI. So this way you can easily group all of the information that you have for your data sets. Like I said before, we are issuing every data set that's included in BIL will get a DOI. But should you need one before that, feel free to reach out to the help desk and we can prioritize that for you. And of course, any questions with DOIs or metadata, feel free to reach out to BIL support and I'd be glad to help you. And I believe that this is actually the end of our presentations today. Are there any final questions, thoughts, or anything related to what we've talked about today? Any DOIs, metadata, anything like that? I will say that tomorrow, if you are coming to tomorrow, we are going to talk a little bit more about how to access data in BIL and how to interact with that data truly. What are the other resources that you have available? I've said before that if you've uploaded data and you need to transform it, what are your options there? What kinds of resources do we have available for you to interact with your data either before you've published it or with other published data sets that are on BIL already? And so we'll go over that tomorrow and some new and interesting things that are happening at BIL. Thank you all so much for coming.
Thanks for the presentation.
Thanks a lot. Very helpful.
Thanks, everyone. All right. I'm going to stick around and answer any questions if you have any, but if not, if you don't have any questions or you don't have any concerns, then I either hope to see you tomorrow or I hope to see your data coming through the Reimage library. Thanks so much, everyone.
So I'm kind of curious about other data transfer protocols besides